Hello and welcome back to another lecture video. My name is Carlos Brian Francia, and today we will be learning about the kidneys. So the kidneys are a very special filter system used by the body, and it's able to filter the blood about more than 200 liters a day. So more than 200 liters of blood and filtrate go through the kidneys every day, and yet we only produce about 2 liters of urine a day. So what happens to the other 198 liters? Well, that filtrate is reabsorbed into the blood, and we are able to reabsorb vitamins, ions, certain carbohydrates, and that's through the usage of the kidneys. And so today we're going to be learning about how the kidney is formed, how it's shaped, and the anatomy of the kidney. On the right side of this kidney, we have a junction, and this junction is called the hilum. So it's very similar to the pulmonary system, in which the hilum is where the arteries and the veins and the nerves, they meet up and they enter the kidney. So that portion is called the hilum, H-I-L-U-M. Underneath this hilum is the renal pelvis, and that's where the formed urine enters, and it eventually goes down and it meets the ureters, and the ureters will carry that urine and push it into the bladder. So this funnel shape right here is called the renal pelvis. This right here is the renal pelvis. If we go upwards, we will notice these weird looking structures, but I want you to kind of focus on this area. Notice how this area is a little lighter than this area right here. So this area that's a little darker is called the renal cortex. It is the renal cortex. It's more of the superficial tissue of the kidneys, and inside here is called the renal medulla. And the renal medulla is where all the ions congregate. It's where all the ions are found, and it's very concentrated in sodium and ions and chloride. And so if we kind of look over here, we will see what I'm talking about. So this portion is called the renal cortex, and this darker portion is called the renal medulla. So this portion is called the renal cortex, and this one's a renal column. But as you go further down, you'll find the renal medulla. If we do a cross section of the kidney, we will notice that the kidney has these lobes, and these lobes right here are called renal pyramids. Usually a kidney will have about 6 or 8 renal pyramids, and inside the renal pyramid is where the kidney takes in water, it reabsorbs ions, it secretes ions. All your diuretics are going to have effects on the renal pyramids. Inside each renal pyramid, you will see nephrons, and these nephrons are actually making the filtrate. So if we kind of zoom in, you'll see the whiteness of this. This is actually one of the loops of Henle, and it's also a collection of those collecting tubules. So like the proximal convoluted tubule, the distal convoluted tubule, they're all found inside the renal pyramid, and there are thousands and thousands of these tubes inside each pyramid, and that's why we can form so much, we can reabsorb so much filtrate every day, and we only make about 2 liters of urine every day, because these guys, these little pyramids, have thousands and thousands of loop of Henle's, collecting ducts, and what have you. And when we talk about those collecting ducts, the collecting ducts, they will take in the filtrate, the quote-unquote filtrate, and they will bring it into this neck right here. So this neck is called a minor calyx, a minor calyx. And this calyx is going to take all that filtrate and it's going to bring it into this large portion. And what did we call this? We call it the renal pelvis, the renal pelvis. So if we hide this fat layer, we will see that all these little minor calyxes will join into this large collection, and this is called a major calyx, the major calyx right there. So the major calyx is going to join into the renal pelvis. So for this model, it doesn't say that this portion is the major calyx, but it is, because all these minor calyxes join into the base of the major calyx, and the major calyx is going to bring all the formed urine into the renal pelvis, and the renal pelvis takes that urine and is going to shoot it into the ureters, and the ureters will meet the bladder. And now the bladder will have that urine and is going to excrete that urine into the environment. If you want to be a baby Einstein and really impress your friends, you can say, yes, we do have renal pyramids, and yes, this is a minor calyx. However, if we hide the minor calyxes, we will notice that each one of these renal pyramids have kind of like these freckles, and these freckles are filters. So now you take this filtrate and you can filter it even more using this filter system. And this filter right here is called a papilla. So that is a renal papilla. So each renal pyramid has another filter inside called a renal papilla. And that's going to allow the filtrate to be even more filtered as it goes down the minor calyxes, and then it's going to be joined into the major calyx, which goes into the renal pelvis. Going back into the renal hilum, when we have the renal hilum, we have the connection of nerves and arteries and veins, but each artery and each vein has a specific name. Now, where do these arteries come from? This artery right here with oxygenated blood, it comes directly from the descending aorta. So when we were learning about the heart, the aorta is like the main artery of the heart. So it shoots out the blood into the aorta, and the aorta carries that oxygenated blood into the systemic circulation. The renal system gets about 25% of that blood from the heart. So they're very perfused with blood. And so this renal artery is a descendant of the descending aorta of the heart. And likewise, the renal vein is going to drain into the inferior vena cava of the heart. When we have the renal artery, we also have kind of like this branching effect. So each one of these arteries are just segments of the major renal artery. That's why we call them segmental arteries. You don't have to remember the name of the segments. Like for instance, this is the superior segment, this one's the middle segment, this one's the inferior segment. It's just location. But just know that we go from the renal artery into the segmental artery. These are just branches of the artery. If we go further along the segmental artery itself is going to have branches, and these branches of vessels from the segmental artery are called interlobal arteries. And what they do is that they're going to go into the lobes of the kidney, and it's going to split even further out. So the segment itself, the segmental artery itself, has segments, and those segments are called the interlobal arteries, and you can find them within the renal columns. So between each renal pyramid is a renal column, and that just separates the renal pyramids away from each other. And notice that we can't really see the arcuate arteries or the cortical radiate arteries because they're really, really small, and they're not shown in this model. And so I have to go even deeper. I have to pull out a microscope for you guys so we can see the vascularization or the vasculature of the kidneys. And so I can show you the rest of this photo. So let's go ahead and let's do that. But before we do that, let's just kind of keep in mind that since we have, for instance, an interlobal artery, we're also going to have interlobal veins. You have these interlobal veins. And so if you have a renal artery, you have a renal vein. You have segmental artery, you have segmental veins. Every artery has a counterpart, and that's called the vein. So oxygenated blood, deoxygenated blood, they're all the same names except you replace artery with vein. If we zoom in even more, we can see that this was once the renal pyramid. If we cut that in half, we can clearly see that inside the renal pyramid we have the renal medulla. So this portion is the outer zone of the renal medulla, and this portion is a really salty portion of the renal medulla called the inner renal medulla. So that is the inner zone of the renal medulla, and it's filled with ions like sodium, chloride, potassium. And if we look in the bottom portion, we can see the papilla. So we have the papilla of the renal system. So that is going to filter this filtrate even more, and it's going to shoot it out into the calyx. So here in the bottom portion we have the minor calyx. Looking at the arteries, we're going from the interlobal artery into the arcuate artery. So this is the arcuate artery. So it's very small and fine, and it gets even more thin. It gets thinner. And eventually it will form these little cortical radiate arteries. So these cortical radiate arteries are going to thin out even more. So when these cortical radiate arteries thin out, they become these arterioles. So these arterioles are going to become even more thin, and they form the capillaries. So in general, in the body, you go from artery to arteriole into capillary. The capillary is the thinnest vessel that has oxygenated blood, the capillary, and it also will slow down the flow of blood so that the solutes from the blood have enough time to diffuse into the interstitial space. So this portion right here, this tissue, is called the interstitial space. So when we're talking about the collecting ducts, when we put water out of the collecting duct into the interstitial space, this is what we're talking about. We're talking about this tissue outside. When the arterioles join into this space, the arterioles become capillaries, and the flow of blood is very slow. This space right here in which they join is called the Bowman's capsule. When we have this formation of the capillaries, this is called the glomerulus. The glomerulus. And you can see that throughout this entire kidney, you have so many glomeruli inside the Bowman's capsule. Now from the Bowman's capsule, you have the pushing of ions and vitamins and glucose out of these capillaries. They get filtered by the Bowman's capsule. There's like a little filtering system right here. We'll talk about that later. And the water also kind of leaves the glomerulus, and it joins into this portion of the Bowman's capsule. It's kind of like a neck. So now you have your filtrate is going to go into the proximal convoluted tubule. And from the proximal convoluted tubule, we call that convoluted because it keeps winding and twisting to increase the amount of time we have to take out solutes from the filtrate. So the filtrate winds and it twists, and as it twists, more and more ions and glucose come out of the filtrate. And it can be modified to add more water or more ions into the filtrate. We go from the proximal convoluted tubule, we're going to go down, 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 and eventually we kind of thin out. And from this portion, we have the loop of Henle. So this right here is the loop of Henle. This wide white structure is called the loop of Henle. So more ions come out, more water goes into the loop of Henle, and in this section, it goes upwards. It goes upwards. And this ascending loop of Henle will meet at the distal convoluted tubule. We call it distal because it has a larger distance away from the Bowman's capsule. So proximal because it's right next to the Bowman's capsule, and distal because this is a little bit further away from the Bowman's capsule. So this portion right here is the distal convoluted tubule. From the distal convoluted tubule, we still have ions and vitamins and glucose or whatever you have being filtered out. And eventually this distal convoluted tubule reaches the collecting duct. So now we have all this filtrate into the collecting duct. You can have water exiting or entering this collecting duct. So this is kind of like the final modification for the filtrate until it reaches the minor calyx and it goes into the renal pelvis, and eventually it forms urine. Throughout the entire filtration system, we have these vessels, and these vessels on the bottom next to the ascending and descending loop of Henle, they are called the vasa recta. And the whole purpose of the vasa recta is to absorb water and is to absorb ions and solutes as they exit the loop of Henle. So for instance, when we have the ascending loop of Henle, we have the outflow of sodium and chloride. And we have a buildup of sodium and chloride inside the interstitial space, and eventually these ions will go into the vasa recta, and the vasa recta will transport these ions into the systemic circulation. If we continue along the vasa recta, we eventually meet another web of capillaries. And these webs of capillaries are called the peritubular capillaries. And they basically do the same thing as a vasa recta, except the placement is different. Instead of being in the medulla, we have the peritubular capillaries inside the renal cortex of the kidneys. And what they do is that they are also going to absorb water, they're going to absorb sodium and ions, they also absorb amino acids and glucose from the filtrate. So for instance, when we're in the distal convoluted tubule and water is being excreted from the distal convoluted tubule, the water will go into the peritubular capillaries, and that's going to bring the water into the systemic circulation, and that helps us regulate the blood pressure and the blood volume, so the amount of water inside the blood. And so the main difference between the vasa recta and the peritubular capillaries is that they are in different portions. The vasa recta is going to be near the loop of Henle inside the medulla, and the peritubular capillaries are going to be in the renal cortex. Before I forget, we also have a collection of vessels right here, and these collection of vessels, they're really just the intrarenal vessels, and the whole purpose of these vessels is not to conduct the filtration process of the filtrate. Instead, it's just to supply oxygen and nutrients to the tissue of the kidney. So the kidney itself is a collection of tissues, and they need oxygen, they need nutrients. And so these intrarenal vessels are just going to vascularize the exterior portion of the kidney, and they're not really involved in this whole process. But you know what is involved in this entire filtration process? Is actually the Bowman's capsule. So we're going to do a deep dive of the anatomy of the Bowman's capsule. But before I do that, I want you to really ingrain this image into your brain. So this is the distal convoluted tubule, and this distal convoluted tubule in this model is kind of far away. It's far away from the Bowman's capsule. In the next model, I want you to realize that this distal convoluted tubule is right next to this Bowman's capsule. So this neck right here is sitting like half a centimeter away from this Bowman's capsule. Not even a centimeter, it's really close. They're like neighbors. And that the reason why is going to be revealed in this deep dive of the anatomy. This is the Bowman's capsule. Another word for it is called the renal corpuscle. And connecting into the renal corpuscle are the arterioles. There are two types of arterioles. There is the entering arteriole, and then there is the arteriole that exits. The entering is called the afferent, that is the afferent arteriole, and this is the efferent arteriole, When the afferent arteriole enters the Bowman's capsule, it goes from arteriole into capillaries, and now the capillaries are all bunched together inside this little confined space, and when they are bunched together, these capillaries are called the glomerulus. Right next to the arterioles are the distal convoluted tubules. So notice that in the previous slide, or in the previous model, the distal convoluted tubule is a little further away from the Bowman's capsule, but I want you to realize that in real life, the distal convoluted tubule is touching the arterioles that lead into the Bowman's capsule. And that's pretty important because there's a whole apparatus, there's a whole mechanism in which the body regulates the filtration. It regulates how fast it goes, how much solute is in the filtrate, and it's all dictated by this collection, this apparatus. Let's say that we have blood entering from the afferent arteriole. The blood goes from the arteriole into the capillaries, and now we have to filter that blood. We have to filter out the amino acids, the glucose, the sodium, the water. How do we do that? Well, the blood is going to be pushed through this weird looking structure, and this structure is called a podocyte. That is a podocyte. And notice that these podocytes have these weird looking red things. Well, these things right here are called pedicels, and they kind of look like ribs, but let's try to find a better picture of a pedicel. Here we have two fantastic pictures. The left picture shows you the connections of the podocyte. So the podocyte are going to form little finger-like structures, and these finger-like structures right there, these are the pedicels. These are the pedicels. And the blood is going to be pushed against the pedicels, and that's going to filter out the blood. So now we have the solutes and water forming right here, and that will go into the proximal convoluted tubule. If we hide the podocyte, we will notice that there is something called the basement membrane. Now the basement membrane is going to be touching the capillaries. If we hide the basement membrane, we will notice that we have the capillaries. So if we hide this, we have the basement membrane. On the right picture, we will notice that the capillaries have these tiny little holes, and these tiny little holes are called fenestrations. And fenestrations, that's just a fancy word to say window. So these capillaries have these tiny little windows. Let's say that we are a sodium ion, and the sodium ion is traveling with the blood. It's going to enter the capillary system, the glomerulus, and is going to enter one of these windows, one of these fenestrations of the capillary. So now we have the sodium leaving the capillary through the fenestrations and is going to meet the basement membrane. It's going to enter this basement membrane. From the basement membrane, it's going to go into the podocyte and is going to be filtered even more through the pedicels. But sodium is so small that it goes through all the filters and it ends up in the proximal convoluted tubule. So let's say we are something larger, like albumin. Albumin is going to have a very tough time going into the filtrate. It's going to stay inside the blood. Why is that? Well, albumin is a very large molecule and is going to not be able to pass the basement membrane. So the albumin is going to try to escape through the fenestration, but the basement membrane prevents it from entering the podocyte and the pedicels. Another thing that you have to remember is that the proteins that make up this lining, they are negative proteins. They are negative proteins. And so particles that are negative, let's say globulins, these globulins are not going to be able to enter the fenestrations because they are negative. It's like getting two magnets with the same side. Positive and positive repel. Negative and negative repel. Because the proteins that make up this endothelium are negative, negative particles are going to be very difficult to enter this fenestration. But particles that are small and positive, like sodium, they are going to be attracted to these fenestrations and they're going to be able to pass through them. And so just remember that the endothelium is made of negative proteins, and so large particles are not going to be able to enter this region. If a large particle does enter, it's usually going to be filtered out by this basement membrane. So that's going to prevent like albumin from entering the solute portion of this section. If you do have albumin or large amounts of proteins, usually there's a hole or a very large tear inside this section, and that could lead to kidney disease. So if you have a large amount of protein inside the solute or inside the filtrate, that is going to show you that you have kidney disease. So there are holes, quote unquote holes, inside the glomerulus that are allowing large particles into the filtrate, when in reality that should never happen. If it does happen, it leads to kidney disease, or it shows you that you do have kidney disease. If we look at the proximal convoluted tubule, you'll notice these structures at the neck, and this is called microvilli. And what it does is that it increases the surface area of the proximal convoluted tubule. Why do we need that? Well, when the filtrate is being formed, there's water and sodium and chloride, and sometimes the body wants those ions back. Sometimes the body wants the vitamins found here to go back into the body. And so we increased the surface area to allow the body more time, more space to absorb those nutrients back. So for instance, let's say we have vitamin D, and vitamin D is in this filtrate. we want the vitamin D back inside the body, and so the vitamin D is going to be in contact with all these microvilli. And the microvilli will allow the vitamin D to go back into the tissue and then eventually into the blood system. So this increases the surface area inside the proximal convoluted tubule. This is just microvilli, and the microvilli are made from cuboidal epithelium. So here we have the microvilli, or the microvillus, made of simple cuboidal epithelium. Once the blood is being filtered, it goes from the capillaries into the efferent arteriole. So we go from capillary into the efferent arteriole. Efferent means exiting arteriole. So this efferent arteriole exits the Bowman's capsule. Now notice that right next to it is called the distal convoluted tubule. And this distal convoluted tubule is very important because it has a sensor. And this sensor that you see here, it's a very compact sensor that senses how much solute is in this filtrate. So if we have too much sodium inside the filtrate, the body will adjust the flow rate. It will adjust how much pressure is being put into this Bowman's capsule. This sensor is connected to the afferent arteriole and the efferent arteriole. And this sensor name is called the juxtaglomerular apparatus. The juxtaglomerular apparatus. We will now be talking about the juxtaglomerular apparatus. The juxtaglomerular apparatus has three different structures. You have the sensor, which is called the macula densa, inside the distal convoluted tubule, and then you have the cells that kind of line the afferent arteriole. That's going to be called the juxtaglomerular cells right there, and you also have the mesangial cells. But first, let's talk about the macula densa. The macula densa acts as a sensor for the filtration system. So this is the macula densa. If we have too much sodium or too much solute inside the filtrate, the macula densa will sense that there is too much solute or too little solute, and it will act accordingly. For this example, let's say that we have a lot of sodium, like we just ate some barbecue and we have a lot of sodium in our blood. Well, there's going to be a lot of sodium inside the filtrate. And when there is too much solute in the filtrate, the macula densa is going to release two signals. And these signals are adenosine triphosphate, ATP, and adenosine. Let's say that this is sodium. There's a lot of sodium inside this distal convoluted tubule. And this is called hyperosmolarity. And that just means that the solute concentration is really high. And it's so high, in fact, that the water portion of this capillary is going to be attracted to this high solute environment. So now you have the water portion of this blood leaving the blood and entering the distal convoluted tubule. We have a lot of water right there. And now that allows the body to urinate more water into the environment. So let's say that you just had a really great barbecue session, so you're eating a lot of sodium. Well, the sodium builds up in the distal convoluted tubule. That pulls water from the blood, and that decreases the blood pressure, and that decreases the blood volume. Blood volume is just the amount of water inside the blood. When you decrease the amount of water inside the blood, there's not a lot of pressure on the wall of the vessels, and that decreases the blood pressure. This also increases urine volume, how much urine is being excreted into the environment. So the body does not like this, because if you keep urinating more water into the environment, you will eventually become dehydrated. There's not going to be enough water inside the blood. Well, what can we do? How can we prevent this? We can prevent this by signaling to the macula densa that there's a high amount of sodium. There's a lot of green ink right here. So the macula densa senses it and they say, we don't want this. We got to signal something. So the macula densa, this guy right here, this collection of cells, the macula densa releases adenosine triphosphate and adenosine, adenosine. And these guys right here, they are going to signal for the mesangial cells. So the mesangial mesangial cells are going to receive the ATP and the adenosine, and they will constrict. Constrict. So now, when these mesangial cells constrict, they will decrease the diameter of the afferent arteriole. So they will push this wall right here closer to this wall, and that narrows the afferent arteriole. When you narrow the afferent arteriole, the amount of blood that can travel decreases. So now there's less blood flow to the glomerulus. If there is less blood flow to the glomerulus, there is less solute being made. Less solute being made equals less urine formation. That must mean that we are not urinating as much. And so the constriction of the afferent arteriole decreases the blood flow to the glomerulus, and that decreases how much filtrate is being made. So before we were having a lot of blood flow, but when we constrict it, only a little bit of blood goes into the glomerulus, and that releases a little bit of filtrate. And so this section right here will decrease the glomerular filtration rate, and that decreases how much water is lost. This buys us some time to get rid of the excess sodium inside the distal convoluted tubule. So over time, the sodium concentration decreases and the water concentration is not as high. So that prevents us from urinating too much and becoming dehydrated. Over time, the amount of sodium decreases. However, we still have to change the tube, because if we keep the tube in this format, we will still have increased water loss. We decreased the amount of sodium here, but now we have to decrease the amount of water inside this section, because if we keep it like this, we're still going to be urinating too much. How do we change it? Well, the macula densa will sense that there is a little bit of sodium. They said, hey, we got rid of all the sodium, now there's only a little bit, so I got to decrease how much constriction we're doing. And again, the macula densa is going to decrease the amount of ATP, and is going to decrease the amount of adenosine. When these guys decrease, the mesangial cells are going to relax. So now we're going to be increasing the diameter. We're going to increase the diameter of the afferent arteriole. So we can say that this guy equals increased diameter of afferent arteriole. If we increase the diameter of the afferent arteriole, that increases the blood flow. So again, we're going to have more blood flow, and that increases how much filtrate we're making. So this will equal an increase in the GFR. But we also have a third player, and this third player are the little juxtaglomerular cells. The juxtaglomerular cells, and here they are right here, so let's kind of highlight them. They're at this section. And what happens is that these guys are going to release renin. And renin is going to increase the vasoconstriction. So this increases the constriction. Why do we need to do this? Well, initially when we had too much sodium, we were pulling water from the blood, and that decreases the amount of water in the blood, and that decreases the blood volume. So initially we had too much sodium, that pulled water from the blood, that decreases the blood volume, it also decreases the blood pressure. And so if we do vasoconstriction, VC, that's going to increase the blood pressure. But now we got to find a way to increase the blood volume. And the way we do this is through renin, because renin eventually is going to lead to the creation of angiotensin 2. It looks like an 11, but it's actually 2. And what angiotensin 2 does is that is going to constrict the efferent arteriole. So the efferent, the exiting arteriole, is going to be constricted. So the angiotensin 2 is going to constrict this portion. But notice that it does not constrict the afferent arteriole. Why is that? Well, we want this afferent arteriole to widen, because when we widen the afferent arteriole, we're increasing the GFR. Now we have more filtrate being formed, but we still want to limit the efferent arteriole. We want this filtrate to build up. If we close this efferent arteriole, there is more time that the blood kind of builds up in this section. So it wants to leave, but it can't really leave. Only a little bit leaves. And so we have this buildup of blood inside this glomerule. And the buildup increases, and now we have more filtrate being formed. So the constriction of the efferent arteriole, it makes sure that the blood stays in the glomerulus, and that increases the GFR. So we can say that renin will increase, we can say it increases the eGFR. Well, why are we increasing the filtrate? Why are we doing that? Well, again, we did this because initially we had too much solute here, and that must mean that we were pulling too much water, which leads to water loss. And so we released renin, which creates angiotensin II, and angiotensin II constricts the efferent arteriole, and that allows the buildup of pressure inside the glomerulus, and that increases the amount of filtrate being created, which increases the GFR. Initially, before we had all this solute, before we had all this sodium, let's say that our GFR was equal to 100. But when we had too much sodium, the GFR was, let's say, 120, which is too high. It's really exaggerating, but let's say that it was 120. So we decreased the GFR by constricting the afferent arteriole, and that allows the GFR to go into, let's say, 60. But when we use renin and angiotensin II, we're going to be increasing the GFR. But that only increases the GFR back to 100. So we go back to baseline. So what I'm trying to tell you is that we do all of this, just so that we can go back to the original GFR, which is 100. We don't want to go past 100. We don't want to do 102 or 110. We want to go back to normal. And the way we do that is that we relax the efferent arteriole and we slightly constrict the efferent arteriole. When we constrict the efferent arteriole and relax the afferent arteriole, we will increase the pressure inside the glomerulus. And that goes from a GFR of 60, which is depressed, and it goes back to the original GFR, which is 100. And so this whole system is just trying to go back to the original baseline. When we go back to the original baseline of 100 for this GFR, the water inside the distal convoluted tubule says, hey, we're regulated, we're normal, let's leave the distal convoluted tubule and let's go back to the blood. So now you have water entering back into the peritubular capillaries, and that's going to increase the blood pressure, and that's also going to increase the blood volume. So angiotensin II increases the blood pressure by constricting the vessels, and it also increases the amount of water inside the blood. So that increases the blood volume. And that is the juxtaglomerular apparatus. The maculodental cells sense the concentration of sodium. They release ATP and adenosine, which constricts it signals for the mesangial cells to constrict the afferent arteriole, and it decreases the GFR. Well, to go back to the normal GFR, we have to relax the mesangial cells. So we decrease the ATP, we decrease the adenosine, and at the same time, we release the renin from the juxtaglomerular cells. The juxtaglomerular cells increase the renin. The renin will make a cascade which releases angiotensin II. Angiotensin II is going to constrict the efferent arteriole. When this happens, this builds up pressure. So at the same time, the afferent arteriole relaxes because the mesangial cells are relaxing, but the angiotensin II constricts the efferent arteriole. So now the blood kind of builds up pressure here and it slowly releases from the efferent arteriole. We have a buildup of filtrate, and this increases the GFR back to the normal rate, back to the normal number. Eventually, the solute concentration goes back to normal, and the water says, hey, we can leave the distal convoluted tubule. Let's go back to the peritubular capillaries, and that increases the blood pressure and increases the blood volume. And this is super important to remember because step questions are crazy on this material. So I'm glad that I could get through it, but that is pretty much the juxtaglomerular apparatus and the tubulo-glomerular feedback system. But before we leave this image, I just want to mention when we have too much solute here and we are increasing the amount of ATP and adenosine, we are constricting the afferent arteriole, but at the same time we're also releasing nitric oxide. So the cells right here, these cells start making nitric oxide, and what this does is that it's going to relax the afferent arteriole. So why are we constricting and why are we relaxing? Well, the mesangial cells are constricting, and they constrict really hard. Sometimes they constrict too much, and we have to really fine-tune how much filtrate we're making. So the nitric oxide at the same time is going to be slightly relaxing the afferent arteriole just to make sure that we're not constricting too much. Because if we have too much constriction, then we have too much decrease of the GFR. And so the nitric oxide is there to really fine-tune how much GFR we're decreasing. So we're not decreasing 50 points or 70 points. No, we're decreasing by 10 points or maybe 5 points. And that's the nitric oxide. It just increases the sensitivity. It increases how finely we constrict the afferent arteriole. Maybe this is your first time hearing about the renin system. So what the Renin-Angiotensin-Aldosterone system is that normally when we have low blood pressure or low blood volume, the kidneys sense that we have low blood pressure and low blood volume, and so renin is going to be released. And renin is made by the kidney. So renin is released by the kidneys, and it's going to interact with something called angiotensin-ogen. So angio just means vessel, and tense means that we're going to constrict, and gen means generation or the creation. So what are we creating? We are creating angiotensin, vessel constriction. So angiotensin is made by the liver, and it's just floating around in the blood. And when angiotensinogen interacts with renin, it creates angiotensin 1. Now angiotensin 1 is flowing in the blood. Inside the lungs, there's the creation of an enzyme called angiotensin converting enzyme, and that goes inside the blood as well. So when angiotensin converting enzyme interacts with angiotensin 1, we get something called angiotensin 2. Well, it looks like an 11, but it's a 2. And what angiotensin 2 normally does is that it's going to constrict the vessels inside the body. So for instance, we're going to constrict the vessels inside the kidneys, and that's going to increase the blood pressure. Later on, you'll find out that we also increase the blood volume. Now, angiotensin 2 also increases the availability of the anti-diuretic hormone, and this also increases, so angiotensin 2 also increases the presence of aldosterone. What aldosterone does is that inside the collecting tubule, the collecting duct, we have these little pumps right here. So these little blue guys are pumps, and what they are is that they are the sodium, potassium, ATPase. So these the little pumps that pump out 3 sodium, and they pump in to potassium per ATP. So 3, 2, 1. 3 sodium out into the interstitial space, 2 potassium inside the collecting duct per single ATP. And as we can see, the collecting duct, so this is the collecting duct, the amount of sodium, actually I like a different color for sodium, the amount of sodium will decrease inside the collecting duct, and remember that the collecting duct is just the urine, and the amount of potassium inside the collecting duct right there, that's going to increase. On the interstitial space, the amount of sodium is going to increase, and the amount of potassium in the interstitial space is going to decrease. Notice that inside the urine, which is this little teal color, the amount of potassium is going to increase over time. And it pulls potassium away from the body. And so this eventually leads to hypokalemia if we just leave it alone. If we continue to use aldosterone, it will lead to hypokalemia, a decrease in potassium. And that's how aldosterone helps to increase the amount of sodium in the interstitial space. But we can't forget about antidiuretic hormone. So how does that work? Well, here we have a separate picture. Let's do the collecting duct right there. We have some sodium right here. Sodium. And at the same time, we also have water. So we have some water right there. Normally, this water would be excreted, and we have a lot of urine. But sometimes we don't want the water to be excreted. Sometimes we want the water to go back into the blood, because if we add water to the blood, we increase the blood volume, which increases the blood pressure. So if we add water to this portion right here, we can see that more water equals more pressure on the walls, and that increases the blood pressure and also increases the blood volume, since the blood is mainly made of water. And so what antidiuretic hormone does is that it's going to insert these little channels inside here. And these are called aquaporins. Aquaporins. And aquaporins are so small that the only thing that can really get into it, is the water. So the water flows out into the interstitial space. And when the water flows out into the interstitial space, it goes back into the vasa recta. Again, the vasa recta are just the vessels that are really near the collecting duct, and they're near the ascending and descending loop of Henle. So the water escapes from these aquaporins and it goes back into the vasa recta. And the vasa recta takes this water and shoots it back into the systemic circulation. And again, that increases the blood volume and the blood pressure. And if we look downwards, we can go back to aldosterone, because I didn't really explain this, but the increase in sodium causes the sodium to build up in the interstitial space, and then it enters the vasa recta. Now we have a buildup of sodium. We have buildup of sodium right here, so we have a lot of sodium. And now the water in the interstitial space, in the tissue, they say, hey, that's a lot of sodium in the blood. Let's go back into the blood. Let's go back into the blood right here. And what this does is that it's going to increase the blood pressure and also the blood volume. So now we know how antidiuretic hormone and aldosterone saves us salt and water, but what does angiotensin II do to the collecting duct itself? So here we have the collecting duct right there, and inside the collecting duct we have some sodium. So we have some sodium right here, and we also have some water. And eventually this is going to form urine. But what angiotensin does is that it's going to activate some pumps. So So here's a pump here, and here's a pump there, right here. And what these pumps do is that they're going to pump out sodium. They're going to pump out sodium, so sodium exits right there. And inside, we will insert hydrogen. So now we have a buildup of hydrogen. So we have a buildup of hydrogen. So the filtrate increases in hydrogen concentration, and the interstitial space decreases in hydrogen concentration. Inside the filtrate, we decrease sodium concentration, and inside the interstitial space, the tissue, we increase the sodium. And again, the sodium will go into the vasa recta, or the vessels, and increase the blood pressure and increase the blood volume by drawing in water. But notice, when you have a large collection of hydrogen inside the filtrate, we are decreasing the pH level. So now this decreases the pH. So maybe we had a filtrate that was normally 6 and now is going to decrease to 4.8. So we have an acidic environment when we use angiotensin itself to use the sodium hydrogen pumps. So angiotensin 2 is the sodium hydrogen pumps. We have antidiuretic hormone that's going to equal the aquaporins. And the aldosterone, that is going to activate the sodium potassium ATPase pump. But all of these guys will increase the blood pressure and increase the blood volume. The blood volume right there. Let's do that. We will now begin our descent into madness. Here at the Bowman's capsule, we are forming the filtrate. But this filtrate has to travel all along the proximal convoluted tubule, go down into the loop of Henle, go up the loop of Henle, and then the distal convoluted tubule, and then finally the collecting duct. So this whole journey will modify the concentration of solutes inside the filtrate. And you'll notice that in the very beginning of this journey, in the proximal convoluted tubule, you have so many modifications happening. So let's zoom in and let's find out what kind of modifications are happening at the proximal convoluted tubule. Before we begin looking at the proximal convoluted tubule, I just want to go back, and I know that this is the distal convoluted tubule, but let's pretend that it's actually the proximal convoluted tubule, because a lot of people get confused about the apical region and the basal region. When we look at the cell, this portion of the cell that is facing the inner section, this is called the lumen section. This portion of the cell that is closest is called the apical region. And the cell that is facing away from the lumen is this portion of the cell. And that is called the basal region, because the basal region is close to the base membrane. It's not really a base membrane, but it's good to remember it as the base membrane, because the basement is the final floor. This region right here, this little membrane, is the final floor for this cell. So this half facing towards the left side is going to be the apical region, and this right side is going to be the basal region. So for instance, in a textbook, it might say sodium is exchanged for potassium in the basal region. Well, that's what it means. Sodium is going to leave the cell going this way towards the right of the screen, and potassium is going to enter the cell going towards the left side of the screen. And the interstitial space, that's just black space right here. But really, in real life, this black space is a bunch of fluid. So again, sodium leaves the lumen. It leaves the inner proximal convoluted tubule, and it's going to leave the cell going towards the right of the screen. It's going to reach the interstitial space, which is the blackness, the void filled with water. And potassium is going to enter from the basal portion, from this portion, and it's going to go into the proximal convoluted tubule, and it's eventually going to reach the lumen. That's this hollow portion right here. So that's just how to read basal section versus apical section. Inside the proximal convoluted tubule, so this is the proximal convoluted tubule, we have so many different modifications. First, you have the export of the sodium going into the interstitial space. When this happens, 3 sodium leave and 2 potassium enter the cell per ATP. So 1 ATP is used to do this. And when this happens, we have a buildup of sodium right here. Eventually, we have a buildup of sodium because the sodium has a high concentration inside the interstitial space, is going to leave and is going to enter the bloodstream. And now the water says, hey, there's a lot of sodium here. I want to enter the bloodstream. This increases the blood pressure and this increases the blood volume. We also have to recognize that every time sodium leaves the cell, it also carries with it a chloride ion. And so now you have a buildup of sodium right here. But in addition to this, you have a buildup of chloride. And if we keep doing this, we have a very large portion of negative ions. And so the interstitial space overwhelmingly becomes negative. And because you want to be a little baby Einstein, when sodium also leaves the cell, it carries with it glucose. So we also have glucose. And glucose is the body's main energy source. So now we're going to have a very large concentration of glucose and also sodium. So glucose is going to be used by the body and sodium increases the blood pressure. But why am I mentioning the chloride ions? Let's find out why. As the filtrate continues down from the proximal convoluted tubule, it is excreting outside right here chloride ions. So we have a bunch of chloride ions being excreted into the interstitial space. Notice that the sodium is being drawn into the blood vessels. There's not as much sodium and there's not as much glucose. But notice that we have an overwhelmingly large amount of chloride. So in the interstitial space, the negativity is going to be increased. When we are going up the ascending loop of Henle, we are shooting out ions. One important fact is that in the ascending loop of Henle, there are no aquaporins. And this is really important because as the ions exit the ascending loop of Henle, water cannot escape. And so the solute concentration of this filtrate is going to be decreased. So the solute concentration of this ascending loop of Henle is going to be decreased as the ions keep moving. Why do the ions keep moving? Well, there are some pumps in the ascending loop of Henle, but notice that we have positive ions and this negative space. So the chloride ions are going to be acting like a little magnet, and they're going to be pulling out the sodium and the potassium from the ascending loop of Henle. And since there is no aquaporin, water cannot escape it, so we become more and more dilute. Dilute meaning that we have a lot of water, but not a lot of ions. So the solute concentration decreases because there is a very large negativity inside the interstitial space that is being created by the chloride ions. And that is the effect of the chloride ion shift from the proximal convoluted tubule. Next up, we're going to be focusing on calcium. So this is going to be focusing on calcium. When the parathyroid hormone is activated, that must mean that in the blood we have a decreased concentration of calcium. So we have a decreased concentration of calcium. When does this happen? This happens when the osteoblast activity is increasing. So they build bone, so they're going to be using the blood calcium to fix the bones, but they're using too much calcium, and so the concentration of calcium inside the blood, inside here, is decreased. We don't want that. We want everything to be regulated. And so parathyroid hormone is going to be increased. And what this does is that it's going to decrease osteoblastic activity, and instead is going to increase osteoclast activity. Osteoclasts are cells that slowly break down the bone and release calcium into the blood. When parathyroid hormone is activated, the calcium inside the lumen, inside the apical region, is going to be shot into the blood. How does this happen? Inside here are these little tunnels, these little channels, and these channels are opened when parathyroid hormone activates them. And now calcium, instead of going into the urine and being excreted, instead calcium is going to say, hey, the body needs me. I'm going to leave the collecting duct, I'm going to leave the proximal convoluted tubule, and instead I'm going to be here in the interstitial fluid. And eventually I'm going to build up right here, and then I'm going to go down into the bloodstream. So now we have a bunch of calcium building up into the bloodstream. Well, now we need this calcium to be transferred away from this area. How do we do that? We're going to do it by using vitamin D. Vitamin D, specifically calcitriol. So calcitriol is hormonal version of vitamin D, and calcitriol is going to create these little binding proteins, So we have binding proteins, and we could think of them as taxis. Now calcium is going to bind to this taxi that was created by calcitriol, which is this vitamin D, and this taxi is going to take it away from this area. And it will take it to a part of the body which needs calcium. And this allows us to avoid being deficient in calcium within the blood. So this prevents the calcium from normally being excreted in the urine. We also have amino acids being pulled into the interstitial fluid within the proximal convoluted tubule. Sometimes the body wants to save the amino acids. For instance, if we're bodybuilders and we want to build muscle, we want to save as much amino acids as we can. And so we pull the amino acid from the apical region, pull it into the basal region, and finally we have a large concentration of amino acids in the interstitial space. Finally, that amino acid buildup goes into the bloodstream, and this amino acid goes into the muscles to make even more muscle. In terms of glucose is the body's main energy source. Again, so we go from glucose from the apical region to the basal region, and this glucose is going to build up in this interstitial fluid and eventually is going to be pulled into the bloodstream. So now we have a large concentration of glucose, and that glucose will go into the cells for energy. For individuals with type 1 diabetes or type 2 diabetes, the amount of glucose in the bloodstream is already elevated. And so glucose can no longer go, it just bounces away from the bloodstream. Type 1 diabetes, there is no insulin production. And insulin is the hormone that grabs the glucose from the blood and it stuffs it into the cell. Because insulin is not present, the amount of glucose in the bloodstream is excessively high. In type 2 diabetes, there is a resistance to insulin. So you are still making that insulin, however, it takes more insulin to drive the glucose into the cells. Again, because there's a high concentration of glucose in the bloodstream, glucose cannot go into the bloodstream, it just bounces away. So we have a buildup of glucose in the cell. Into the proximal convoluted tubule. And because there's a large portion, we go downwards. So right here we have the filtrate. We have all the filtrate right there. So eventually this filtrate will become urine, but now you have such a large quantity of glucose that the urine is going to have a large concentration of glucose. And when there's a large concentration of glucose, water is going to be pulled to the high area of solute. So now we have so much water being pulled that there is more solute right there. So there's more urine being formed. And eventually, that is going to make a diabetic urinate even more. So we have a large amount of urine being formed, and that leads to dehydration. So that's why people with diabetes, they may feel thirsty all the time. We have polydipsia. This is polydipsia. When you feel thirsty all the time because you keep urinating. And when you keep urinating, that is called polyuria. And that is when the diabetic person keeps urinating and now they feel thirsty. because they are dehydrated. The reason why is because there's so much glucose in the blood that the glucose just goes into the proximal tubule and eventually goes into the filtrate, which eventually becomes urine. So now the urine is very sweet. It tastes sweet and it also smells sweet. Not like I've actually tasted it myself, but the urine smells sweet and it has a high concentration of sugar. And this ruins the kidneys, because since you have so much solute, the GFR is always increased. The higher the GFR, the more filtrate we're making. The more filtrate we're making, the more urine we're making. Since the diabetic has such a high concentration of glucose inside the urine, the GFR is going to be increased, and it's like you're stepping on a gas pedal. You can't do it forever, and so now the kidneys, they start crashing, and that leads to chronic kidney disease. That's when the glomerulus is strained, and now there are tiny holes inside the glomerulus. Now we're dealing with magnesium, and we're also dealing with water. Both magnesium and water, they can easily pass from the apical region into the basal region, and from the basal region they go into the interstitial space, which eventually leads to the bloodstream. But what about PO4 or phosphate? What about this guy? Well, PO4, 3- phosphate, this is called phosphate. What happens here is that it wants to go into the bloodstream, and when it enters the bloodstream, it's going to combine with the hydrogen to form hydrogen phosphate. And the reason why we form hydrogen phosphate is to buffer the blood. So we want to regulate how much hydrogen is inside the blood. If there's too many hydrogens, then the blood becomes acidic, and we don't want that. If there is too little hydrogen in the bloodstream, we become basic or alkaline, and we also don't want that. And so we have this phosphate acting as a buffer. But you know what else is a buffer? Bicarbonate. So we have HCO3 minus, so we have a minus right there. So this right here is the bigger buffer. So we use bicarbonate even more than we use phosphate. The reason why is because the product of this guy, so if we have a hydrogen adding to the bicarbonate, we get water, and we also get CO2. And CO2 is a gas, and it can be exhaled easily. And also the concentration of this bicarbonate is a lot higher than phosphate. So phosphate is not really used because there's a lower concentration in the body, but we always want to use the bicarbonate as a higher concentration, and also we get a gas that can be excreted via exhalation. And that can also be regulated inside the proximal convoluted tubule. When we are forming the filtrate, some of that filtrate has bicarbonate. Now the body doesn't want to get rid of the bicarbonate that easily. We want to save this bicarbonate because it's a really important buffer system for the blood. If we get rid of the bicarbonate, then the concentration of hydrogen inside the blood is going to increase, and that makes the blood acidic, so the blood will be a pH of maybe 5, and that's a little too acidic. Instead, we want to be around like 7.2. So we have to find a way to save this bicarbonate, because bicarbonate doesn't cross from the apical to the basal cell easily. It doesn't do that easily. It needs help. So what we're doing is that when we're drawing sodium into the interstitial space, we're pumping the sodium from a low concentration into a high concentration. So there's already a high concentration of sodium. So now we're pushing this low concentration sodium into the interstitial space. And when we do that, we are pushing hydrogen into the basal membrane, and that uses ATP. So this is an active pump. Why are we pulling hydrogen into the basal cell? Well, we're pulling hydrogen because we want the hydrogen to diffuse into the apical region, and that allows this hydrogen to combine with the bicarbonate. So here's what we're doing. We get this bicarbonate and we combine it with hydrogen. When we do that, we're going to be using an enzyme called carbonic anhydrase enzyme. And the product of this is going to be carbonic acid, H2CO3. But that only lasts for like half a second. Carbonic anhydrase enzyme again is going to break down the carbonic acid and is going to release water and carbon dioxide. water can just go from the apical membrane, from the basal membrane, and then into the interstitial space. Easy. It doesn't need any help to do that. What we really wanted was this carbon dioxide, because carbon dioxide is a gas. It can flow easily from the apical region into the basal region. So now we have carbon dioxide here. And some of this water, again, is going to join the carbon dioxide. And now we have carbonic anhydrase again creating carbonic acid. And again, carbonic anhydrase is going to break this down to make hydrogen and also the bicarbonate. So we were being a little sneaky because we can't move bicarbonate from the apical into the basal by itself. No, we need all this process so that we can move the bicarbonate into the basal section. So now we have this bicarbonate here, and the bicarbonate can go into the interstitial space, and from the interstitial space it can go into the bloodstream. So we have the HCO3, this is the bicarbonate, and now when it combines with this hydrogen, it's going to form water and also carbon dioxide. And that's going to decrease, it's going to decrease the amount of hydrogen inside the blood, and it's going to increase the pH into the 7 range, into the normal healthy range. And that allows the body to avoid metabolic acidosis. So metabolic acidosis is when there's too much hydrogen, and that's going to decrease the pH. And that's really bad for the body because that blood is way too acidic. This bicarbonate buffer system allows the body to avoid metabolic acidosis. However, if we keep doing this and we don't regulate it, then eventually we go from 7.2 into a higher pH. So let's say maybe a pH of 8 or 7.6. Well, that is too basic for the blood, and that leads to metabolic alkalosis. So that's when we do this process of combining bicarbonate with hydrogen and getting rid of the hydrogen concentration. So that drives up the pH to around 7.6 or 7.8, and that's a little too basic for the blood. And if you keep doing it, you have metabolic alkalosis. I also want you to realize that when carbonic acid splits into bicarbonate and hydrogen, some of this hydrogen doesn't pair with the bicarbonate in the apical region. Instead, some of this hydrogen is used to pump out different solutes. For instance, some of that hydrogen is used to pump out sodium or chloride or calcium or amino acids, and it's all used as a recyclable product. So we recycle that hydrogen product and we're going to use it to pump out even more solutes. So the body is very sneaky and it's very smart. There are certain medications that can actually inhibit the carbonic anhydrase enzyme. So the carbonic anhydrase enzyme inhibiting medications, they are going to act as diuretics. So what is a diuretic? Well, a diuretic is a medication that will increase the urine volume. So they will cause you to urinate more liquid. And when we urinate more liquid, we're getting rid of the water inside the bloodstream. And that decreases the blood pressure, and it also decreases the blood volume. So what carbonic anhydrase enzyme inhibitors do is that they prevent the formation of carbonic acid. So now we have a buildup of bicarbonate ions. And remember that bicarbonate cannot move from the apical region into the basal region. And now we have a buildup of bicarbonate ions. And now bicarbonate is going to be pushed into the filtrate. So on this side, right here on this side, we have filtrate being formed. Since there's an increase in bicarbonate, the bicarbonate is going to move into the filtrate. So we have a lot of bicarbonate. Since the concentration of bicarbonate is increased, the water is going to say, hey, I like an area with high solute, so I'm going to move into this section. So all the water is going to be pulled into the filtrate. So that increases the amount of water that is in the filtrate, and that increases the urine volume, and that increases the amount of urine being formed, which leads to a lot of water being lost in the urine. So that makes us urinate more. However, we don't really prescribe carbonic anhydrase enzyme inhibitors to increase the urine volume, because this is really weak. We don't really increase the urine volume when we use these types of medications. So why do we prescribe them? We prescribe them in the treatment of glaucoma. So in the treatment of glaucoma. And glaucoma, that's just an increase in blood pressure inside the eye. Inside the eye is a fluid called humerus. And if you have more humerus inside the eye, the humerus will pull, or it will push on the walls of the eyeball, and that's going to increase the blood pressure. And that causes a loss of vision over time. The reason why we can prescribe carbonic anhydrase enzyme inhibitors is because bicarbonate, this guy, is partially forming the humerus. So when the humerus fluid is forming, it uses bicarbonate. So if we can build up the bicarbonate and excrete it out, we can decrease the glaucoma. We can decrease the blood pressure in the eye by decreasing the amount of humerus made. And that's why we would prescribe carbonic anhydrase enzymes. It's to treat glaucoma. And so a lot of these medications are actually found in eye drop forms. Another reason why we might prescribe carbonic anhydrase enzymes is because there might be too much bicarbonate concentration in the bloodstream. So when hydrogen combines with bicarbonate, it will increase the pH. So we go from a pH of 7.5 into something that is slightly more basic, like 7.6. However, if there's too much bicarbonate in the bloodstream, it might lead to metabolic alkalosis when the blood is too basic. And so we can prescribe carbonic anhydrase enzyme inhibitors to decrease the amount of bicarbonate in the bloodstream. That will go from a pH of, let's say, 8.2 all the way down to maybe 7.3 or 7.5, something that is healthy. So we can prescribe it for glaucoma, and we can also prescribe it for metabolic alkalosis, though it's not that common. And a precaution, if your patient has an allergy to sulfur, the medications that are carbonic anhydrase enzyme inhibitors, typically they have a sulfur in them. And you don't want to prescribe it to a patient that has a sulfa allergy. So sulfa allergy, beware. Here are some carbonic anhydrase enzyme inhibitor medications. Notice that a lot of these medications have zolamide. So acetazolamide, methazolamide, brenzolamide. When you see zolamide, you should be thinking of carbonic anhydrase enzyme inhibitors. And typically they're in eye drop form, so you would give an eye drop of acetazolamide to a person who has glaucoma. You have your generics on top, and you also have your brand names on the bottom. Moving past the proximal convoluted tubule, we now enter the loop of Henle. So here is the loop of Henle, and notice that we have two portions. We have one portion going downwards from the proximal convoluted tubule, and we have another portion that carries the filtrate going upwards. Because one portion, the left portion, is going downward and the right portion is going upward, we call this an opposite flow. So this is a counter current exchange. As we go down in the descending loop of Henle, we will lose water. And when we lose water, the concentration of solutes increases. But how are we losing water? Well, outside in the interstitial space is something called urea. And urea is a breakdown of ammonia. When we are digesting proteins, let's say you just had 3 protein drinks, the body will digest that protein, and a byproduct of protein breakdown is ammonia. So ammonia is going to be broken down, into urea. Why do we do this? Well, ammonia itself is toxic. and that will hurt the body if the concentration of ammonia is increased. And so we want to convert it quickly into urea. Now urea is going to be building up in the interstitial space, and because there's a high concentration of urea, water likes the high concentration, and it's going to go out into the interstitial space, and eventually it will go into the bloodstream. So the water will go into the blood, increase the blood pressure, increase the blood volume. But urea is going to go from a high concentration into a low concentration. So the urea, as we go down in the descending loop of Henle, the urea is going to start building up. So when we're losing water, the concentration of the solute increases. So we go from 300 to 500 solutes to 700 solutes to 900 solutes, because urea keeps going into the descending loop of Henle. On the right portion of the ascending loop of Henle, so we have the ascending loop of Henle, we will notice that there is no water leaving. The reason why is because there are no aquaporins. And this portion of the loop is made from cuboidal epithelium. So cuboidal epithelium. This portion right here is made from squamous epithelium. So that is just simple squamous epithelium. Well, why is this important? Well, it's important because if we don't have any water leaving and we have ions leaving, we're going to be decreasing the solute concentration. And as we keep going upwards, we're going to be increasing the amount of water that remains. So if we have 5 milliliters of water right here, we're still going to have 5 milliliters of water as we go up. But if we have, let's say, 1 gram of ions here, as we continue going upwards, eventually we only have like 0.2 grams of ions. And so technically we have more water compared to ions as we keep going upwards in the ascending loop of Henle. Now remember that chloride is building up inside the interstitial space because of the proximal convoluted tubule. As we build up the chloride in the interstitial space, we're going to be using them as a magnet to draw out the sodium, to draw out the potassium. When the sodium and potassium leave the ascending loop of Henle, they also carry chloride ions. And that's a little special. Why is that? Well, the way that it leaves the ascending loop of Henle is because it uses these little channels right here. And these channels have special names. These channels are going to excrete 1 sodium, 1 potassium, and 2 chloride ions. We have 2 positive ions and 2 negative ions. So the 2 positive ions are going to be neutralized by the 2 negative ions. So there is no gain in positivity, there is no gain in electronegativity. And scientists are really good at naming things, and so they named this the NaK2Cl channel. That's all it is. There's 1 sodium being excreted, 1 potassium being excreted, and 2 chloride ions being excreted out of the ascending loop of Henle. Now we have medications that can prevent this from happening. And these medications are also diuretics. And these guys are called your loop diuretics. Diuretics. What a loop diuretic does is that it's going to block this membrane channel. It's going to block the excretion of sodium, it's going to block the excretion of potassium, and it's going to block the excretion of chloride. So here we are, we're going to plug it up. So now sodium can't leave. It's trapped. So now the concentration of sodium is increased. And what happens if we have an increased amount of sodium? Eventually, when this filtrate reaches the collecting duct, so here we have the collecting duct right there, we have a very large amount of sodium, we have a large amount of potassium, and we also have a large amount of chloride. And since we have so much solute, the water inside the interstitial space will say, hey, I like areas with a lot of solute. I'm going to go and I'm going to join the filtrate, and I'm going to increase the amount of water that is being lost in the urine. So when I take a loop diuretic like Lasix, that's going to make me urinate a lot. And if I keep taking Lasix, I eventually become dehydrated because I keep urinating all the water outside. However, we have to realize that inside the collecting ducts, we have more membrane channels. So here we have a membrane channel, and again we have another one. And these guys are special because this one is going to be a sodium potassium ATPase. And what this does is that it will take the increased sodium and is going to move the sodium into the interstitial space. So the concentration of sodium is going to be decreased. But when this happens, it will shoot the potassium into the filtrate. So the amount of potassium eventually increases. So now we have a massive potassium loss, and that leads to hypokalemia. That's when we have a decrease of potassium inside the bloodstream. And so what I want you to learn is that if we keep taking Lasix or loop diuretics every day, and we don't replenish your potassium levels, if you don't take Micro-K, which is a potassium medication, it just inserts potassium into your body. If you don't take that, eventually you become hypokalemic, and that could lead to heart problems, and we don't want that. And you're probably wondering, well, Brian, where is chloride? I thought there was chloride here. Yeah, there is. When sodium leaves into the interstitial space, chloride also follows it. So it will follow sodium into the interstitial space. So while you do lose sodium and chloride and potassium when you use loop diuretics, the most critical ion that you have to pay attention to is potassium, because potassium will actually be lost easily. Because look, 3 sodium leave the collecting duct and 2 potassium enter the collecting duct. For every sodium that leaves the collecting duct, you have more potassium entering. When you have 3 sodium leaving the collecting duct, you have 2 potassium entering the filtrate. So eventually that leads to massive potassium loss in the urine. So that leads to hypokalemia because you have 3 sodium that goes into blood, and you have 2 potassium that is leaving the blood to enter the filtrate, and that is per every ATP. So 3, 2, 1 ATP. And it is this loss that leads to hypokalemia. Here are some examples of loop diuretics that you may encounter in the hospital. So you might encounter bumetanide or ethnocrinic acid. The brand names are Bumix and Edicrin, but you will always encounter furosemide and you will always encounter torsemide. So furosemide is actually Lasix, and torsemide is also known as Demodex. Now, one thing that you have to remember is that torsemide, which is Demodex, is twice as strong as Lasix, and it also lasts longer inside the body. So for instance, if we're giving 20 milligrams of Lasix, which is furosemide, that is equal to 10 milligrams of torsemide or Demodex. So we have Demodex. And Demodex, which is torsemide, is going to last longer in the body because it has a higher half-life. So this guy right here has a higher half-life. And again, loop diuretics, they just inhibit the excretion of ions from the ascending loop of Henle. That is why they're called loop diuretics. When we go past the ascending loop of Henle, we enter the distal convoluted tubule, and that is this section right here. So this is the distal convoluted tubule, and it's a lot easier to understand conceptually. The main thing that you have being excreted into the bloodstream is sodium and chloride. When sodium leaves the distal convoluted tubule to enter the bloodstream, chloride is also going to leave with the sodium. When we have something called aldosterone, and you should remember him, aldosterone is going to increase the amount of channels inside here. So specifically, we have these little membranes that we're going to insert right there. Let's enter him too. And these guys are actually the sodium potassium ATPase. So again, they're going to shoot out sodium from the distal convoluted tubule, and it's going to build up inside the interstitial space. When we have a large amount of sodium in the interstitial space, eventually it goes into the bloodstream and it pulls water into the bloodstream, and that increases the blood pressure and also the blood volume. When this happens, potassium goes from the interstitial space back into the distal convoluted tubule, and we have a large concentration of potassium right there. And that gets excreted into the urine, and if we keep doing that, it leads to hypokalemia. But this is without any drugs. This is without any medication. When we leave the distal convoluted tubule, the sodium builds up in the interstitial space, but so does chloride. So always think of chloride as being the twin brother of sodium. Sodium chloride together, that makes salt. So when sodium leaves, chloride also leaves. But what if I want to use a medication to prevent sodium and chloride from leaving the distal convoluted tubule? What kind of medication would I prescribe? Well, I would prescribe something called thiazide and thiazide-like. And thiazide-like just means that it's a medication that acts the same way as a thiazide drug does, but it has a different structure and it doesn't interact with the body as a thiazide does. So it's kind of like a little cousin of a thiazide. Anyways, these drugs are going to prevent the secretion of sodium and chloride. But how do we do that? Inside the distal convoluted tubule, we have this little channel. And these channels are not the sodium-potassium channels. No, these guys are going to be the sodium-chloride channels. So sodium and chloride, they go from a high concentration into a low concentration, and so naturally, they just want to flow out. So this guy flows out, and we now have sodium and chloride into the interstitial space. And this happens a lot. So you can see it happening there and there. Thiazides are going to say, we're going to block you out. We're going to block you. So we're going to put a cover on these channels, and now sodium can no longer leave the distal convoluted tubule. And chloride can also not leave the distal convoluted tubule. So now we have this buildup. We have this high concentration of sodium and chloride. When we reach the collecting duct, we have a lot of the sodium-potassium ATPase channels. So we have sodium right here. We have this channel. So again, think about the loop diuretics. When we have a large concentration of sodium right here, we have a large concentration of sodium, they're going to leave because this is a different channel. Right here, this is a completely different channel from the sodium chloride channel. This is a sodium potassium ATPase channel. So 3 sodium leave, but when this happens, you now have 2 potassium entering right there. And again, you also have some chlorides also leaving with the sodium. But now you have this high concentration of potassium, and that gets excreted out of the body via the urine. So again, this leads to hypokalemia. So you have to be very careful when you're prescribing diuretics to a patient, because over time, the potassium levels is going to be depleted. And so typically you would give micro K, which is just a capsule of potassium, and you give it sometimes to a patient. So for instance, if I'm using like hydrochlorothiazide and I'm using it for a long time, or I give a high dose, I would give micro K alongside with it. So hydrochlorothiazide is a thiazide diuretic, and it's going to prevent sodium and chloride from leaving the distal convoluted tubule. So you have to be careful with hypokalemia. Here you have your thiazides and your thiazide-like medications, and you have the brand names. The most famous thiazide is going to be hydrochlorothiazide, so that is actually HCTZ. And you can often see HCTZ paired with blood pressure medications. For instance, ACE inhibitors like Benicar. You can have a combination of Benicar with hydrochlorothiazide. You can also have thiazide-like medications taken by themselves. For instance, I've seen several people use chlorthalidone by itself, and their blood pressure goes down and they urinate more. So in general, you'll almost always see chlorothiazide and hydrochlorothiazide when you're doing your rounds in hospitals. You'll also see chlorothalidone. Indipamide, you don't really see it, but sometimes people do use indipamide as a thiazide-like medication. In general, some people state that thiazide-like medications will decrease the blood pressure, but they also decrease the risk of having hypokalemia. But just know, when you're giving diuretics to a patient, you always want to measure or monitor the amount of potassium in their blood, and you also want to monitor the amount of chloride and sodium in their blood. You don't want them to be hypo anything. And in general, you don't want to be giving diuretics to a person with chronic kidney disease. For instance, somebody on dialysis where they have stage 5 renal disease, so the GFR is like below 20, below 15. You don't want to do that because that's going to put too much strain on the kidney, and it also increases the risk of being hypokalemic. So people on dialysis, they have an issue balancing their potassium levels. They might have too much potassium, they might have too little potassium. You don't want to be giving them diuretics. So always be on the safe side with a person with kidney disease. You don't want to be giving them diuretics. You don't want to stress out the kidneys even more because the kidneys are already failing. So don't give them diuretics. Just monitor their potassium levels. As we go from the distal convoluted tubule to the collecting duct, normally we have some solute right there, and that just goes and it goes and it goes into the urine, and that's just in a normal fashion. Inside the collecting duct are again the little channels that are sodium, potassium ATPases. So normally we have some sodium, and the sodium is going to go into the interstitial space, some of the sodium, while some of the sodium stays inside the collecting duct. With the sodium, you also have your potassium. Without any medication, some sodium goes into the interstitial space, and some potassium enters the collecting duct from the interstitial space, and together they form the urine that will be excreted. So that's in a normal fashion. However, there are some medications that prevent the sodium from leaving, and they also prevent the sodium from exiting in the urine. How do we do that? How do we build up sodium, and how do we prevent potassium from entering the collecting duct? The type of diuretic that we're going to use is going to be called a potassium sparing diuretic. So diuretic. And what this does is that it's going to block the sodium potassium ATPase. So normally sodium wants to exit. Well, we say and we block these sodium potassium ATPase channels. So now sodium cannot exit the cell. They cannot exit the collecting duct. So we're going to bounce. Here's the sodium. We're going to bounce out. So we cannot exit the collecting duct. Likewise, the sodium that is the potassium that is outside right here, it cannot enter the collecting duct. So the potassium cannot enter the collecting duct right there. So slowly we have a buildup of potassium in the interstitial space, and that will decrease the amount of potassium that is being lost in the collecting duct. Because sodium cannot leave the collecting duct due to the blockage of these potassium sodium ATPases, we have an increase in sodium. So now we have an increase in sodium, and that draws water from the interstitial space into the collecting duct, and it's going to increase the amount of urine. So this increases the urine volume, but it also takes away water from the blood. So we decrease the blood pressure, and we also decrease the blood volume. Now, potassium-sparing diuretics, they also decrease the effects of aldosterone. Because remember, aldosterone is going to increase the activation of these sodium-potassium ATPases. Now, potassium-sparing diuretics is going to decrease the effect of aldosterone. So that's just going to increase the concentration of sodium inside the collecting duct. That's going to pull water from the interstitial fluid and is going to prevent the entrance of potassium into the collecting duct. So that increases potassium overall. And you also have to be careful with this because this could lead to hyperkalemia, which is when you have too much potassium in the blood. And since you have such a loss of sodium, that might lead to hyponatremia, which is too little sodium. So we are losing the sodium from the blood, so that leads to hyponatremia. However, potassium-sparing diuretics are kind of weak when they're used by themselves. So typically you use potassium-sparing with loop diuretics. So you want to use them in combination with Lasix or torsemide. So you can use potassium-sparing diuretics with loop diuretics. Here are some potassium-sparing diuretics that you may see. So you'll always be looking at spironolactone, and the brand name for spironolactone is called aldactone. Aldactone. So aldactone, so ALDACTONE, aldactone is going to inhibit the sodium-potassium ATPase channels. Another one that is kind of gaining popularity is finerenone, which is called Corindia. And in the clinic that I interact with patients, there is a drug representative for Corindia, and this drug representative is trying to sell this medication to individuals with chronic kidney disease. And the risk that you have with chronic kidney disease is that you run the risk of having hypokalemia, which is a decrease in potassium. Because this is a potassium-sparing diuretic, it's actually going to increase the potassium levels. So that's also dangerous for the patients. But Corindia is used for individuals that have like stage 3 chronic kidney disease and not stage 5. You don't want to be using diuretics for individuals with a very, very bad chronic kidney disease. But stage 3 is okay. It's not going to do that much harm. In fact, you don't want to be doing any harm to the patient. But Corindia is used to prevent the exacerbation or the worsening of this chronic kidney disease. So that's kind of like the exception that you would give it to a person with chronic kidney disease. That would just be Corindia. But you never give diuretics to a person using dialysis. And typically that's like stage 5 chronic kidney disease. And the final diuretic that we're talking about is going to be called osmotic diuretics. Diuretics. Now there are two types of osmotic diuretics. There's called mannitol, which is kind of like a sugar, and the sugar is not absorbed or excreted by the nephron. Instead, the sugar builds up and it just goes all the way over here and it leaves the distal convoluted tubule, and it actually leaves the collecting duct. But because there's such a high concentration of this sugar, the water in the interstitial fluid is going to be drawn into the tubules because there's such a high concentration of this sugar. And so overall, the amount of water is going to increase, and that's going to leave as urine. So mannitol is given via IV, and it just increases the amount of water that we excrete because this mannitol cannot escape from the tubules. The reason why you would give mannitol is for somebody who has a lot of swelling inside their head, specifically intracranial. This is intracranial hypertension or swelling. So there's too much swelling inside the head. You would give mannitol via IV, and that increases the amount of water that is being sucked into the tubules, and overall you're going to be urinating a lot of water. Another medication that we can give, and it's more typical, is called isosorbide. And that can be called osmotic, or it can be called isobide. Mannitol, the brand name for mannitol, is actually called osmitrol. And typically you only give osmotic diuretics if you have intracranial hypertension or swelling, or if you have a lot of pressure inside the eyes. So you have swelling inside the eyes, like intraocular hypertension. And the way that it enters is that it's going to have an effect on the proximal convoluted tubule and also the descending loop of Henle. So we have loop of Henle. And actually isosorbide is given more. So I've seen people use isosorbide to lower their hypertension for their blood pressure. So you'll see isosorbide in a tablet form, and you'll see it more often when you're in the hospital. And here are your 5 diuretic classes. You have loop diuretics, you have thiazide and thiazide-like diuretics, you have potassium-sparing diuretics. These guys are going to be working with almost every day. And you're also going to be having carbonic anhydrase inhibitor diuretics and osmotic diuretics. So hopefully now you know what the diuretics do, some examples of those diuretics like the brand names, and the warning for these diuretics. We will now be talking about the effects of hormones on the blood pressure. Recall that when mesangial cells sense ATP and adenosine, they will constrict, and that decreases the diameter of the afferent arteriole. This decreases the GFR. But at the same time, there's a hormone being released, and it's released by the endothelial cells of the arteriole, and this is called endothelin. And endothelin also increases constriction. And that's going to further decrease the GFR. And when we decrease the GFR, we decrease the flow of blood here, and that decreases the amount of filtrate, and that gives the body enough time to absorb the sodium. The heart can also regulate the blood pressure via hormones. So when the blood pressure is elevated, the atria increases. So the atria swell up with all this influx of new blood. So when this happens, the heart is going to release a hormone called atrial natriuretic hormone. So atrial natriuretic hormone. And what happens here is that atrial natriuretic hormone is going to decrease the amount of aldosterone, it's going to decrease the amount of antidiuretic hormone, and that will decrease the amount of sodium that is reabsorbed into the body. So if we decrease aldosterone production and we decrease antidiuretic hormone, we will decrease the amount of sodium that is being reabsorbed into the body, and we will decrease the amount of water that is being reabsorbed into the body. When we do this, we decrease the blood pressure and we decrease the blood volume. Therefore, the atria do not swell up as much. They swell up when we have too much blood pressure, and so we release atrial natriuretic hormone to decrease aldosterone, we decrease the aquaporins from the antidiuretic hormone, and therefore we decrease the blood pressure and the blood volume. When we decrease antidiuretic hormone, we increase the amount of urine that we urinate out. When we decrease sodium-potassium channels, we increase the amount of sodium inside the collecting duct, which will draw in water from the interstitial fluid into the collecting duct, and thus it will also increase the amount of urine that we lose, and it also increase the amount of sodium that we lose. So overall, atrial natriuretic hormone decreases sodium in the blood, it decreases the water amount in the blood, and therefore it decreases blood pressure and blood volume. There is also another type, it's called B type, the letter B, and it's a natriuretic peptide. However, it's made by the ventricles, and the ventricles don't really produce it as much, and when it does produce it, it is weakly affecting. So it is going to do the same thing as the atrial natriuretic peptide hormone. but it is weaker and it is secreted by the ventricles, so we don't really pay attention to it. We really pay attention to the atrial natriuretic peptide hormones. So now we can take a deep breath and relax because we finished talking about how urine is formed. Now we just got to talk about how it's excreted. And the way it's excreted is that once the urine is formed in the kidneys, so if we hide this portion, the urine is going to collect right here in the minor calyx. So the collecting ducts reach into the minor calyx. It goes into the renal pelvis. And from here, the renal pelvis converts into the ureters. So this guy is called the ureter. And the ureter is made from smooth muscle. And the smooth muscle will contract via the autonomic nervous system and uses an action called peristalsis to push the urine into the bladder. And if you're upside down, it can still push the urine into the bladder because it doesn't need gravity. It's using the smooth muscle to contract. Inside the ureter, we have a special type of epithelial cell called the transitional epithelia. And what it does is that it allows the epithelial cells to kind of stretch like an accordion. So when there's a lot of fluid, the epithelial cells will stretch to accommodate all that extra fluid. So instead of being this wide, we can have ureters that are a little bit wider and a little bit shorter. So it will vary as the amount of urine is produced. So if we have a lot of urine being produced, the transitional epithelial cells will allow the ureters to expand or to contract. Here we have the bladder. So the urine goes from the ureters into the bladder, and you will notice that the bladder has some ridges, and this is called the detrusor muscle. And what the detrusor muscle does is that when the bladder fills up with urine, the detrusor muscles, which is smooth muscle, will start to contract. As we expand the bladder, we increase in size, but the detrusor muscles don't like that, and they want to go back into the original shape. When this happens, it gives us the urge to urinate. A simple concept is like taking a rubber band and stretching it. You feel that pulling force that is opposing your hands. So when you stretch the rubber band, you can't stretch it forever because it wants to go back to the original shape. And that's what's happening in the bladder. The more urine we have, the more contractions we have in the bladder, and that gives us urgency to urinate. So if I drink 16 gallons of water for some reason, I'm going to have a very large bladder and it's going to start contracting. That means I want to go urinate. And if that feeling is overwhelming, then sometimes a person urinates in their pants, and that is called incontinence because the bladder can no longer hold that much water. But how do we urinate? How do we have that urination reflex? Well, to urinate, we're going to be using 3 different types of nerves. The first one is it's going to be called the pudendal nerve. The second one is going to be called the hypogastric nerve. And the third one is called the pelvic nerve. And all 3 of them are going to be working together to make that urination reflex. When the bladder fills up with urine, the detrusor muscles will stretch, and when they stretch, they will send signals to the parasympathetic nervous system, and the parasympathetic nervous system is going to relax the internal sphincter, and that relaxes the muscles here, and that allows the urine to flow from the bladder into this region. Now, the parasympathetic nervous system again is going to send more signals, and that's going to inhibit the motor neurons of the external sphincter, and that allows the urine to travel from this area into the urethra, and that exits into the outside environment. When we have that urge to urinate and we hold it in, quote unquote, hold it in, and we squeeze, when we squeeze that external sphincter muscle, we are using the pudendal nerve. So we're going to be using that pudendal nerve for voluntary control of the external sphincter, and that allows us to hold our urine in. The pudendal nerve arises from the sacral micturition center. That's just a fancy word to say urination center. Micturition just means urination. Since we can hold our urination via the external sphincter muscle, we can say that we have somatic control over the external sphincter muscle. And therefore, we can say that the external sphincter muscle uses acetylcholine for contractions. So the external sphincter muscle for the urethra is going to be using acetylcholine for contraction. And that allows us to hold our urine. when we are being chased by a bear or a chihuahua, we don't want to urinate. Why is that? Well, we have the sympathetic nervous system taking control of us. We use the hypogastric nerve. So the hypogastric nerve is just part of the sympathetic nervous system. And you can think of it as hypogastric, like low stomach activity. When you're being chased by a bear, you don't want to eat anything. You're not hungry. You're not digesting food. So you have a low gastric activity. So you have the hypogastric nerve. That's just an easy way to remember it. In this case, the hypogastric nerve is just going to decrease the urge to urinate. However, if we have so much urine in the bladder, the parasympathetic nervous system is going to decrease the sympathetic signals from the hypogastric nerve. So it will override the signals created by the hypogastric nerve, and you're going to urinate anyways. Now that we know how urine is formed and what the GFR means, we can actually put it into clinical practice. So here we have a person's urinalysis. So this is just a test that analyzes the urine. And we will notice right off the bat that the glucose level, the amount of glucose in the blood, is elevated. So it's elevated at 108. So that just shows that we have some glucose retention in the blood. We also have something called creatinine. And creatinine is just a byproduct of protein breakdown. Every day we have proteins breaking down and we have proteins being rebuilt. Creatinine is just a byproduct of that protein breakdown. If you have elevated creatinine levels, then the kidneys are not filtering the creatinine. Normally, the creatinine is less than 1.4. There's a range. If we have elevated levels of this, then the kidneys are not functioning well. So you can look at this and you can say, hey, we have a level of 7.39. That must mean that my kidneys are not filtering well. That must mean that I have a depressed GFR level. And if you look further down, we notice that the estimated glomerular filtration rate is depressed. So we have a GFR of 6. And that's just a range of the filtration level. If we have a depressed level, then you have chronic kidney disease, and that leads to renal failure. So here we have a severe case of renal failure. This person would definitely be on dialysis. And therefore, if you're on dialysis, I would not recommend diuretics, because this person is prone to hyperkalemia. And look, we have a level of 6.9 for the potassium. That itself is hyperkalemia. We have hyperkalemia. And if I give diuretics, then this is going to get even worse. Even if you're thinking, hey, diuretics, they lead to hypokalemia and we shouldn't worry about it. No, don't even think about diuretics when you're dealing with a person with this level of eGFR. Don't even think about it. Their kidney is destroyed. They need a new kidney transplant. They need dialysis. We cannot afford diuretics. We can't even do Corindia, which is a potassium-sparing diuretic. We can't do that, because if you mess with the potassium, it just goes all the way down to the trash. So hyperkalemia, hypokalemia, whatever you do, just monitor the potassium levels of this patient. Don't even think about diuretics, because the kidneys cannot tolerate it. We have an eGFR of 6 that is severely damaged kidneys. So the creatinine level is elevated, and that must mean that we don't filter the creatinine levels as well as we should. And that gives us an idea of how damaged the kidneys are. And then if we reveal the eGFR, we can see that, hey, we have a severely depressed kidney. Here is a chart from the National Kidney Foundation, and we can see the eGFR ranges. So normally you want to be above 60 or higher. So everybody eventually reaches stage 2 kidney disease. It's not really kidney disease. Like, you may have a 30-year-old who has a eGFR of 88. They don't have that disease. However, as you age, you lose more and more of your kidney function. So for that individual who had an eGFR of 6, well, that is severe. That is severe kidney failure. We actually label it as stage 5 chronic kidney disease. So you might see stage 5 chronic kidney disease. And that is very common in people who have type 2 diabetes. You have so much glucose going into the kidneys that over time it ruins the filtration system. It ruins the filtration system. And over time the eGFR decreases from, let's say, 60 all the way down to 15 or below. And typically you need dialysis, like stage 4 and 5, for sure they need dialysis, but stage 5 definitely needs a renal transplant. So we have different ranges. We have stage 3A, stage 3B, stage 4, stage 5. Most commonly, you're going to see individuals with stage 3A chronic kidney disease. So that's usually between 45 and 59 eGFR. And this is a chart of the average eGFRs between the years. So I'm 22 years old right now, and I should have an eGFR above 100. As you age out, when let's say you're 70 years old, you should have an eGFR above 70. For the most part in real life, you'll see a lot of patients with eGFRs of 50, 65, and they're doing well. You only see the effects of chronic kidney disease when you get into depressed levels, like eGFR of 30 or 41. That's when they start having a lot of symptoms for their kidneys, like edema or high potassium or low potassium levels in the blood. So we just finished looking at a basic metabolic panel, but now let's look at the urinalysis. So the urinalysis is the analysis of the urine. And here we have this table. So we can see that this person's urine was yellow and it was kind of cloudy, so we might be thinking infection. And we have the specific gravity. So specific gravity is the amount of solute inside the urine. If we have a high specific solute or a specific gravity, we have too much solute and we're not filtering as well as we should. Leukocyte esterase, that's an enzyme given off by the white blood cells. So when the white blood cells see a bacteria, they will kill the bacteria and they will release enzymes called leukocyte esterase. So notice that we actually have some leukocyte esterase in the urine. So that should give us a warning. It should be like a red flag. Hey, for some reason we have white blood cells in the urine. We're fighting some sort of bacteria. I'm thinking infection. You should be thinking that. Sometimes, though not all the time, you will have the presence of nitrites. So bacteria, specifically gram-negative bacteria, they are going to eat the nitrates. So nitrate is normally in the urine. So we have nitrate being in the urine. If you urinate right now, you'll have nitrate inside your urine, but bacteria will eat the nitrate. Get it? They ate the nitrate and they convert the nitrate into nitrites. When you have the presence of nitrites, that should be another red flag that you are dealing with a urinary tract infection. So the presence of nitrites will sometime lead to infection. So if you look at nitrites and you see positive, you're like, oh, we may be dealing with an infection, though it doesn't happen all the time. Like for instance, we have an infection here, but we don't have the presence of nitrites. We also have the pH, and the pH is in a set range. The presence of proteins inside the urine, it will tell you, hey, for some reason, the glomerulus is not filtering the protein. So we have some damage inside the glomerulus, and we have this excess amount of protein inside the urine. So that's not good. The same thing is true with glucose. Maybe too much glucose is leaving the glomerulus and is entering the urine. So that could be a sign of kidney damage, though not all the time. For instance, you may have a diabetic who is pretty young, like a kid. Maybe he's like 10 or 12, and he just has a lot of glucose in his blood because he's a type 1 diabetic. Ketones, that's the energy of the body. So, for instance, typically we use glucose for the energy, but if we're on a keto diet or we're not eating a lot of carbohydrates for whatever reason, the body switches from the carbohydrates and it switches to the breakdown of fats. So when you break down fats, it increases the amount of ketones. Ketones is just a byproduct of fat metabolism. So the presence of ketones shows you that the body is not using glucose. You will often see this within diabetics. For instance, a type 2 diabetic, they don't have enough insulin, so they can't push the glucose into their cells. And therefore, the glucose is just in the blood, and it's not being used by the cells for energy. So instead, the body uses fat for energy, a little bit of fat, not too much, and it will increase the amount of ketones in the body. Urobilinogen can be used to kind of dictate how much heme breakdown we have. So the same thing is true for bilirubin. If we have these guys that are elevated, that says, hey, for some reason we have heme breakdown. So for some reason, I am breaking down my red blood cells. I'm breaking down my erythrocytes. That's not good. If I have too much breakdown, I have too much bilirubin, and that increases. If I can't filter out the bilirubin, it stays in the liver and it can actually cause jaundice. But bilirubin and urobilinogen, they should be kind of like red flags. For some reason, there's some heme breakdown, and I got to figure out why the body is breaking down the erythrocytes. You're really going to be using the bilirubin. Urobilinogen just makes urobilin. Urobilin, if I can spell it right. And that leads to urochrome. And urochrome just gives you the yellow color in your urine. You also have your red blood cells, the white blood cells, the epithelial cells, but notice that we have some bacteria inside the urinalysis test, inside the urine. So there is some presence of bacteria. I wonder what kind of bacteria we have. Here you have the urine culture. So this is the urine culture. And this urine culture typically takes about 7 days to get back to you because they're growing the bacteria from the urine. And you would order a urine culture to an individual that you know will have a lot of UTIs. So for instance, this person keeps coming into the clinic and they're elderly and they say, hey, I keep urinating, my urine burns, and I have an odor in this urine. Also, I'm confused. Can we get some antibiotics? And you say, hold on, let me order a urine culture first before I order some antibiotics. The reason why you do this is because you want to know what kind of bacteria you're working with, and you want to give the best type of penicillin or other medication. So sometimes it's not even penicillin. Sometimes it could be something like Cipro in this case. So in this case, we have this specific bacteria, and we see that is gram negative. And we also have these numbers. So these numbers will tell you the best antibiotic to give. So for instance, we have an antibiotic that has the number 1 and is also sensitive. Therefore, this number 1 would be better than this number 2 and this number 4 or number 8. So in this example, let's say that we have no allergies and we're dealing with somebody who can tolerate Cipro. So I would give Cipro 500 milligrams twice a day. That's twice a day BID for 10 days. That's assuming that this person does not have an allergy to Cipro and they can tolerate this dosage. However, if we have an individual who has impaired renal filtration, so maybe they have stage 3 or stage 4, well, I don't want to give them the full dosage. Instead, I give them a dosage of 250 milligrams twice a day. So twice a day for 10 days. And that's a reduced dosage. So maybe you're an older person, maybe you're dealing with an elderly person or a person who has some renal impairment. You don't give them the full dose of any antibiotics, you give them the half dosage. What if we have an allergy to Cipro? Well, we wouldn't use Cipro, we would use a different antibiotic. So that's why we run this panel. If you don't have this option available to you, you can pick out different options from this list. And we can also see what kind of medications are being resisted. So for instance, ampicillin is a type of penicillin and it's resistant. So this bacteria is not gonna be killed if I give ampicillin, so I'm not gonna prescribe it. Sometimes in real life, all the good antibiotics are resistant and the only good antibiotic is sensitive, but it's only in IV form. So ideally you want to give something that is in the mouth, like Cipro. Cipro can be PO, that's a tablet form. However, there have been occasions where I've seen that patients are resistant to all the antibiotics except for one antibiotic, and that is given IV. For that case, we would either send them for an inpatient facility, such as a hospital, and you give them that antibiotic. However, a lot of people don't want to go to a hospital. They don't want to go to a hospital, so you would go into an outpatient facility. For instance, like home health. These are the nurses that go to your house. They have the ability to give you IV antibiotics, and they go to your house and they are monitored by a doctor, and they give you that antibiotic. So you see a lot of people who are elderly will only respond to IV antibiotic. So they have the option of doing inpatient or outpatient. Inpatient means you go to the hospital. Outpatient means you get that service, but not inside the hospital. So maybe inside your home. So it really depends on the urine culture. What kind of antibiotic would be beneficial to your patient? What kind of antibiotic doesn't trigger an allergic reaction? What kind of antibiotic doesn't hurt the patient? Ultimately, you do not want to give an antibiotic that hurts anybody. You just want to give the best care possible because you love your patient. You treat them like family. Here's a great example of a urine culture that shows a high resistance to antibiotics. In this case, the patient did a urine culture on Monday and they did not experience relief, so they did another urine culture, and this culture just came in. So this culture shows a high resistance to almost all of the antibiotics. Initially, in the first urine culture, the bacteria was sensitive to Cipro, so we gave Cipro, but the bacteria adapted, and now the bacteria is almost resistant to everything. The only thing that's not resistant is tobramycin and amikacin. But notice that we're going to be giving tobramycin instead. Why are we giving tobramycin and not amikacin? Well, assuming that your patient doesn't have any allergies to the specific antibiotics, tobramycin has a greater sensitivity. We have a sensitivity of 4, and 4 is better than 16. So we want to use antibiotics that are closer to the number 1. If I had an antibiotic that was number 1, we would pick that antibiotic. So for instance, if we did not have that resistance to Cipro, it's really close to number 1, so we would rather use it. However, we're resistant to Cipro, so we're not going to be using Cipro. So in general, use an antibiotic that is either 4 or below. So 4, 3, 2, 1, these are good numbers to have. Using an antibiotic that's maybe a 6 or an 8, that's not that helpful. I wouldn't really give that antibiotic. You also need to realize something important. These antibiotics, amikacin, tobramycin, gentamicin, you're going to be seeing these guys a lot. And what is special about them is that they are the old school antibiotics. These guys were made in the 40s, and they're called aminoglycosides. So these are aminoglycosides. And what's special about aminoglycosides is that if you have an excess amount of aminoglycosides in your body, they can harm the ears. So if we have an infection inside the ear and we give tobramycin in the form of Tobradex, Tobradex can be given to people with pink eye, that's conjunctivitis. They can be given to people who have ear infections, that's otitis media. You can give Tobradex, and Tobradex is just a combination of tobramycin and a steroid called Decadron, or dexamethasone. We could just say Decadron. But anyways, when you give these medications, you have to be careful with what you're giving it with. So for instance, if I have these aminoglycosides and I give a loop diuretic, this is going to be a little harmful because the loop diuretic increases the concentration of aminoglycosides in the ears, and that causes ear damage. So you would worry about that in patients who have renal insufficiency. So a person who is perhaps stage 4 chronic kidney disease, they would be really sensitive to the concentration of aminoglycosides. In normal individuals, like for maybe a 30-year-old or a 40-year-old who's really healthy, you wouldn't necessarily worry too much about it. But for somebody who has renal insufficiency, you would worry about the aminoglycoside concentration. So be careful with the loop diuretic. What do you do in this case? Well, you still need that antibiotic, but instead of using it every day, we use a small dosage. We use maybe a half dosage. We use a half dosage on Monday, Wednesday, and Friday. And that just decreases the risk of having hearing damage as a result of using aminoglycosides and loop diuretics. For this patient, tobramycin is not easily accessible in oral form. We have to give it in IV form, so we have to do it intravenously. And typically you would send a patient to go to the hospital. However, this patient had a physician who is specialized in bacteria and diseases. That's called an infectious disease physician. And so that physician was able to get that patient into the office and monitor the IV administration. So that physician actually did IV tobramycin for that patient, and now they are doing very well. So sometimes you can give IV inside the office of a specialized physician. For instance, it was the infectious disease physician, and this patient did not have to do inpatient care in a hospital. Instead, they did outpatient care. And that is a great example of resistance to antibiotics in a urine culture. Now let's kind of wrap up things that I may have forgotten to mention. So the first thing is caffeine. When you drink tea, coffee, soda, those typically have caffeine, and caffeine is going to be acting as a diuretic. And the way it does it is that it's going to increase the vasodilation of the glomerulus. So that is going to increase the GFR. So when we are dilating that afferent arteriole, caffeine helps us do that, and that increases the amount of filtrate we're making, which increases the urine volume, which increases H2O loss. When we drink 4 shots of vodka, we're drinking alcohol. And alcohol is also going to be acting as a diuretic because it inhibits antidiuretic hormone. So we decrease the amount of aqua porins, and that increases the urine volume, which increases water loss. So when you drink your shot of vodka, about 10-15 minutes later you need to use the restroom because you are inhibiting antidiuretic hormone. The inner lining of the bladder is made from transitional epithelial cells, and what the transitional epithelial cells do is that they allow the bladder to expand, and when you expand, you accommodate more liquid. So if we didn't have these transitional epithelial cells, the bladder would be very hard to expand with urine. And these transitional cells, you can imagine them as accordions. So the accordion is really compact. It's really compact. But when it expands, it becomes like this, and it can hold more volume. In the heart, we have atrial natriuretic peptide, and in the ventricles, we're going to have the beta type natriuretic peptides. And what these guys do is that we're going to increase the sodium concentration in urine. That's going to increase the urine volume and increase the water loss. That's going to decrease the blood pressure and decrease the blood volume. But for the B-type natriuretic peptides in the ventricles, that is going to be weak. So this guy is weak. However, the half-life is large. And so it might be weak, but it works over a longer period of time. And so we can use this as a diagnostic marker. If we have a large concentration of B-type natriuretic peptide, if we have a large concentration of this in the blood, that can act as a red flag for heart failure. And so sometimes you might see doctors ordering tests for this concentration because it can show that the heart is doing very badly. It can show heart failure. And that is it for this video. So hopefully now you understand how the urine is formed, how to read an antibiotic chart, how to look at a urine culture and a basic metabolic panel, and hopefully you learned something. So I'm glad that you spent your time learning with me, and I'm glad that you watched this video. So remember that you are awesome and you are super cool. So I love you so much, and I wish you the best. I hope that I gave you a great education. Thank you so much, and I love you. Bye.